So now our first topic is quantitative MRI for atherosclerotic plaque, presented by Maria Altbach from the University of Arizona. Welcome. Okay, so I go forward.
Hi, good afternoon. So I guess multi-contrast MRI provides unique information on plaque composition, and you've heard some talks before, and the idea is that some of these plaque components are associated with plaque vulnerability, so it's really it's an area where everybody's really interested in trying to get this type of information from the vessel wall. Currently, in really most vascular surgeons, they really just use lumen to determine when they want to operate or not. And in many cases, the lumen doesn't really provide that information. It's really in the vessel wall. Now, the approach of using multi-contrast MRI, the conventional way is to compare signal intensity of the plaque components to the adjacent muscle using a combination of time of flight, T1 weighted, T2 weighted, and proton density weighted images. And this is a table taken from the literature. And you can see how, do you have a pointer? Is this the...
Yeah.
Okay. Let me see. Oh.
Here, how about this one? This one.
All right, thanks. All right. So you can see that different sequences here provide different contrast for these different components of the plaque. So, and this is another set of images taken from the literature to illustrate how these are three different types of plaques and with different types of contrast. And you can actually see, if we just even look at the T1 weighted and T2 weighted images, you will see that the combination of those two will just give enough information to be able to differentiate, for instance, a lipo-rich necrotic core without intraplaque hemorrhage, or one that has either a fresh IPH or a recent IPH. So that actually and this is actually has been going on for many years, but it's just more recent that people start thinking in using quantitative techniques, T1 and T2 mapping, to provide information on plaque characterization. And that should be more accurate in terms that things could be quantified and it could be done directly. So it really will not require necessarily a radiologist that have a specialized experience in being able to determine what this plaque is based on contrast. Hopefully it will be done automatically. So the requirements for techniques for plaque characterization for doing quantitatively is to have an adequate number of images for parameter fitting. If you want accuracy, you need to keep the spatial resolution similar to what is currently done in the conventional multi-contrast. You need good anatomical coverage, motion robustness. I think you've heard in different talks before talking about that. And you want to do this in a reasonable amount of time, maybe 3 to 5 minutes. And so with that in mind, there's been work already by different groups in developing T2 and T1 mapping sequences primarily, but there's also work in diffusion-weighted MRI and dynamic contrast enhancements and T2 star mapping. And I'm just going to talk a little bit about these. So here's an example taken from Chai et al., where they image a subject. So this is in vivo imaging, and then they obtain the specimens and do histology. And you can see this is an area here of lipid, and in the T2 map, that shows as a low, have a fairly low value. Next to it, there's an area of intraplaque hemorrhage, and that has, a high value in T2. So that's actually show pretty, nice promise that, this T2 mapping could distinguish between these different components in a plaque. Now, if you only do T2 map, then, in this case of, if you have a lipid-rich necrotic core without IPH and a recent IPH, you could do a very nice, it could distinguish those two. But if you have a fresh IPH, you couldn't do it. But if you add, T1 mapping, now you have a nice combination of both, and then you could differentiate, the three of them. And, the reason for that is obviously there's changes in the cellular composition of fresh IPH and recent IPH, which is, it's really translated into differences in T1 or T2. So, and this is what, people have done for T2 mapping. You will find papers using, the conventional multi-echo fast spin echo techniques or T2 prep gradient echo. For T1 mapping, there's papers on variable flip angle methods or the ones with the inversion recovery flash, which is a loop locker technique. More recently, these combined methods, and you've heard these before, this multitasking approach where you can get T2 and T1 information from a single scan. These things have been developed for 2D and 3D, and also Cartesian and non-Cartesian. And the non-Cartesian methods, you heard before, like radial MRI will provide robustness to motion, but I think more importantly, it provides the ability to, radial, you're frequently sample the central part of K-space, which allows high temporal resolution compared to Cartesian scanning. So, and this is the work that we've done. We have these T2 and T1 mapping radial MRI sequences. For T2 is a 2D radial turbospin echo sequence, which we collect 12T images to provide very accurate signal modeling, allowing for imperfections like stimulated echo, and in a fairly short imaging time. And the T1 mapping technique is the 2D radial loop locker, produces 40 TA images with bright blood contrast, and in about 2 minutes. And one of the things, the reason why we can go fast is because radial provides, has really very strong robust properties for undersampling, so we don't really need that many lines for each of these contrast images, the Ds and the TI. Sometimes we use 4 to 5% of what's determined by the Nyquist condition. And we recover the rest through, compressed sensings or AI reconstructions. So another good thing of multi-contrast data, obtaining these parametric sequences, is for vessel segmentation. That's another topic that was brought up before. So for instance, here from the T2 data, this is from our data, our black blood images have a very nice anatomic delineation of the plaque. So that helps us define the outer wall, whether we want to do it manually or you want to use it with a segmentation algorithm. For the lumen, actually, it's much better to use one of the TI data sets, the one at TI of 450. Remember, we collect 40, so it's easy to get one that we like. And, it helps really delineate the lumen very well, and then you combine both, and you get a nice mask that you could readily apply to the T2 and T1 map. And so that actually points to the ability to eventually automate these things for the user, the end user that really doesn't have much experience of delineating things manually or even want to do it. Another thing that's really good of these multi-contrast T2 and T1 weighted images is the delineation of calcium. So calcium will have a signal void both in T2 and T1 weighted images. This is a case of a plaque, and you can see these void here. It's also seen in the T1 weighted image that's obtained, as part of this parametric exam. And if we compare these with the time of flight, you can see it. You can verify that that is calcium. And in fact, there's even further verification with histology. This is another case where actually the area of suspected calcium is very close to the lumen, so sometimes it's very hard to tell if you only look at the T2 weighted image. But if you look at the corresponding T1 weighted image that was acquired in this set, then you can very clearly see where's the lumen and where's the calcification. Again, in comparison, this is sort of verified with a conventional time of flight and also with histology. So with all that information, you can have a better picture of what's going on in the plaque. And here's one example of somebody with a fairly complex plaque. Looking at the histology, there's clear presence of calcium, but also intraplaque hemorrhage, and it has a quite high T2, and kind of an intermediate T1. So that is consistent with a fresh IPH. This is another example of somebody with an intraplaque hemorrhage. In this case, T2 is low, and T1 is really much lower than that one. So that is consistent with the presence of a fresh IPH. And this is a case of an intraplaque of somebody which has a lipid-rich necrotic core, but not intraplaque hemorrhage, but there's clear lipid here, and has a lower T2 value, but T1 is really not much affected. So in this way, you can actually have a nice picture of the plaque from these maps. This is the work, actually, you've seen these images before, but you'll see it again, from the QMatch sequence developed by colleagues at Cedars. That's this 3D T2IR continuous flash method that provides bright blood for lumen characterization, the dark blood for the wall, and then the T1 and T2 map with obviously the corresponding T1 weighted and T2 weighted images that are used to generate those. That's in about a 7-minute scan. In their work, they also have seen, that the combination of T1 and T2 is really important to characterize these components. And here's work from data from 48 subjects, where you can see that depending on the plaque component, things are clustered in this T1, T2 space. Moving away from T1 and T2 mapping, there's diffusion-weighted MRI also, and this is a slide that was shared by our colleagues at University of Utah, and they developed the 3D stack of stars sequence. And with that one, they were able to show that when you have an intraplaque hemorrhage that is very clearly seen, like here in the MP range, the ADC is low. And more important than that is that they were able to look at what does it mean in subjects. So you can see that the ADC can separate between symptomatic and asymptomatic subjects, and more importantly, like here, the ones that have IPH versus the ones that don't. Another technique that's another paper that's been in the literature that I thought it was pretty interesting is the quantification of T2 star. This group used a multi-echo gradient echo sequence, and they acquired the data in 34 subjects and compare in vivo results with endarterectomy. And they analyzed actually the tissue with mass spec to look at total iron, and also with electron paramagnetic resonance looking at iron-3 compounds. And what they found is, well, first is that the symptomatic patients have a shorter T2 star compared to asymptomatic patients. When they looked at the total iron content, it was interesting that there wasn't really much difference. There was no significant difference in terms of total iron content. But there was a clear difference in the amount of iron-3 species. And the ones that are here that are sensitive to EPR are these low molecular weight iron-3, and it is less in the symptomatic patients, which led them to conclude that in here, most of the iron is based on the high molecular weight iron-3 species like ferritin or hemosiderin, which are known to have a low T2. So this is quite interesting because it really can tell you a lot more about the nature of the composition of these blood products. Another technique that has been used is dynamic contrast enhancement. And this group had looked at kinetic modeling of the DCE in plaques, looking at the fractional plasma volume, VP, and transfer constants, K-trans. And here you can see that when they correlate the data derived from in vivo against the analysis of the ex vivo tissue, there was a nice positive correlation between the presence of macrophages, both in VP and K-trans, neovasculature, and also loose matrix. So there's really, in general, in DC, there's quite a lot of development in improving this technique to have higher anatomical coverage and high temporal resolution. So it just shows promise as a way to characterize inflammation in the vessel wall. So in summary, quantitative mapping provides really a new way of analyzing the vessel wall characteristics. The information that can be obtained from different parameters can be used to assess the presence or absence of plaque components, and more importantly, hopefully the plaque vulnerability. There's more work needed to assess reproducibility, to validate this against the more conventional methods and then against histology, and to standardize the methods such that everybody can use. I mean, the hope here is that you would have a technique that automatically, that basically will acquire data that's quantitative. You can automatically segment the wall, or there will be, a software that will do it and can actually classify the plaques without really the need of these very experienced radiologists to do it. It is really important also to develop algorithms that can combine this information from these various parameters to interpret what this means. And the two major goals is to provide a reliable method for assessing risk of stroke and to understand the effect of therapeutics in plaque progression and regression. So with that, I like to acknowledge investigators at the University of Utah and Cedars-Sinai for providing slides and funding. And I just want to point out these two sources of funding has been sort of recent funding of a multi-center project. We are 6 institutions, and you will hear more about it on Fridays, a talk by Dr. Parker. All right.
So